doctor Nelson Brown es médico, egresado de la Pontificia Universidad Católica. Posterior a eso se muda a Alemania, donde hace el doctorado en medicina, y posteriormente se mueve a Boston a hacer un postdoc en el tema que luego siguió desarrollando aquí en la Universidad de Talca, que es cáncer y ciclo celular, senescencia.
Hola, hola, hola. Ya.
Bueno, yo personalmente quiero agradecer al doctor Brown por estar acá, por presentar en esta ocasión, y también porque, bueno, yo les decía en la mañana, cuando este programa se acreditó, bueno, obviamente todo el comité académico participó de ese proceso, pero sin lugar a dudas, sin el apoyo del doctor Daniel González y del doctor Nelson Brown, eso no habría sido posible. Así que pido un aplauso por el doctor.
Thank you very much for staying so long. Today I would like to done during the last 3 or 4 years. There are many things that I will show that are already published, so I will be quick and I will go through those. So probably you know this slide very well. So we are experiencing global aging of the population worldwide. Only in Chile, there's an increase, or there was an increase in the life expectancy of about 30 years since the 1950s. And this demographic transitions means that there has been an increase in the incidence of chronic diseases, including cardiovascular diseases and cancer. In fact, cancer is the main, aging is the main factor, or one of the main factors involved in the development of the most common types of cancer, which are epithelial in origin. Research focus on aging is therefore very important for us. So here is a definition of aging. Aging is the progressive loss of physiological integrity as a result of accumulated cellular damage. This cellular damage can be caused by many, or can be associated to many mechanisms. In fact, there are hallmarks of aging which have been described over the years. For example, genomic instability, telomere shortening, alterations in stem cells, etc., etc. One of these hallmarks of aging, which is very important for us, is cellular senescence. In fact, there's accumulated evidence that cellular senescence is very important for organismal aging. That is, as we age, we accumulate senescent cells in several of our tissues. But what is cellular senescence? Well, this is a short definition. It's a unique form of cell cycle arrest which can be triggered by in response to a wide range of physiological and pathological stimuli. In fact, it's a phenomenon that was described almost 60 years ago by these two men, Leonard Hayflick and Paul Moorhead. Basically, what they observed was that when fibroblasts, primary fibroblasts isolated from humans, were propagated in culture, they stopped dividing after some number of doublings, and they defined that as a Hayflick limit. We know now that cellular senescence is a process that limits the proliferation of primary human cells in culture, and it is caused by the progressive shortening of telomeres. This process was originally called replicative senescence, and soon after it was described, it was clear that cellular senescence could function as a tumor suppressor process. In other words, to be transformed or to be fully transformed, a cell that on its way to become cancerous must overcome this process of cellular senescence. As a corollary of this idea, mutations that activate pathways that are involved in the orchestration of cellular senescence are important or are mutated in most human cancers. And among these pathways, you have the P53 pathway, of course, and the RB or retinoblastoma pathway. So in 1997, it was discovered that replicative senescence is not the only type of senescence that exists. There is something called stress-induced senescence, also called premature senescence, which means that the same phenotype can be produced by a number of stimuli, including drugs. Yeah, many of the drugs we use in chemotherapy, for example, to treat patients with cancer, we know now that are inducing senescence and not cell death or apoptosis. And among these compounds or drugs that are pro-senescence are a number of cytotoxic or originally considered cytotoxic drugs, but also targeted, new targeted drugs like the ones we use in our lab. One of them called palbociclib, which is an inhibitor of cycle-independent kinases that act at the point of progression from the cell cycle, from the G1 phase of the cell cycle to the S phase of the cell cycle. Senescent cells are not any type of cells that just exit the cell cycle. They have some features, very specific features. For example, they are increased in size, they are flattened when we study them in culture. They show multiple vesicles in the cytoplasm, and they, very importantly, they increase the amount of this beta-galactosidase activity at pH, particularly in senescent cells of human origin. But most, or more importantly for us, and far from being simple bystanders or passive bystanders, are very actively producing and secreting a wide array of factors, including growth factors, cytokines and chemokines, components of the extracellular matrix, and metalloproteinases. Yeah? In other words, senescent cells, even though they don't proliferate, they can remodel the tissue microenvironment by secreting all these factors that can have very pleiotropic effects, both locally and physiologically as well. So we can consider senescent cells, in fact, as inflammatory cells, and many propose this hypothesis that the accumulation of senescent cells in an aging organism basically produce this low-level pro-inflammatory context that can produce alteration. One reason is because platelets are very well studied in our university. There are very strong groups working on platelets, and at some point we decided that an easy way to go doing research in this university was combining some of our research lines. So most of what I'm going to show you in the next slides was done in collaboration with Rodrigo Moore and Claudio Valenzuela, who have pushed this line of to develop cardiovascular diseases. So the simplest hypothesis at the time was that factors that were secreted by senescent endothelial cells could lead to platelet activation. Yeah? So remember that activation and aggregation of platelets is a very stereotype kind of process. It begins with platelet adhesion, platelet activation, which is characterized by changes in the shape of platelets and the secretion of the several factors that are contained in these secretory granules. But the activation is finished by the platelet aggregation, which is, of course, the end results and the factor that promotes primary hemostasis. So, program Alexandra Olate-Briones, who is also a PhD candidate and is working now with Dr. Herrada in Universidad Autónoma, Claudio Valenzuela, Angel Cayo, all important people who have contributed to the data I'm going to show you now. So the experiment we designed at the beginning was very simple. So we took advantage of this human microvascular endothelial cell, which is an immortalized endothelial cell line. And we basically start to induce senescence through several stimuli. Finally, we decided to use doxorubicin, which is a chemotherapy drug, to induce senescence in the cell line. We use basically 15 nanomolar of this drug to induce senescence. We have then our model set, and we check. Well, in this case, when you study senescence, the first thing you have to do is beta-galactosidase activity. Took advantage of other markers. One of the main markers we use in the lab is T21, which is an inhibitor of CDK, but also one marker which is a negative marker for senescence, in the sense that senescent cells downregulate this protein, is laminin B1, which is reduced in senescent cells. So we were at some point pretty sure that what we are seeing is really senescence. So the next step was characterize these senescent cells in terms of the SASP, in terms of the phenotype, the senescence-associated secretory phenotype. So we selected a number of factors that we knew could be produced and secreted by senescent cells, but and could also have an influence on platelets. And we observed that many of these indeed were upregulated at the mRNA level in senescent cells. We then tried to determine the presence of many of these factors, but now in the conditioned media derived from the cells. We observed that, for example, which is one of the factors that at the mRNA levels were seen very highly upregulated, was also present in conditioned. We observed that using the conditioned media derived from senescent endothelial cells, these media were able to induce more adhesion compared with the medium derived from non-senescent cells. So it was clear that platelets were able to be were more adhesive when they were influenced by conditioned media derived from senescent cells. As part of the process of platelet activation, we also saw some important changes in the size of platelets, which are typical of platelets that are undergoing an activation process. And we saw that this activation process was particularly important when platelets were exposed to media conditioned by senescent cells. We also observed that many of the markers for activation of platelets were also upregulated in host platelets that were exposed to conditioned media derived from senescent cells, from senescent HMEG1 cells, which are the endothelial cells we are using. Yeah? But in this case, many of these experiments, as you can see here, many of these experiments only were evident when we use sub-aggregating concentration of this agonist, which is ADP. Yeah, but nevertheless, we observed that platelets that were exposed to conditioned media derived from senescent endothelial cells were capable of activating, which is what we expected. But finally, we, of course, had to prove that platelets exposed to conditioned media derived from senescent cells were able to aggregate, and this indeed was what happened. So these experiments, although simple, demonstrated, I believe that we demonstrated for the first time that factors released by senescent cells would indeed induce platelet activation and aggregation. So that was in the context of senescent cells of endothelial origin. However, we were also interested in how senescent cells would influence tumor progression. You can imagine that, for example, in the context of chemotherapy, the accumulation of senescent tumor cells could lead to a pro-tumorigenic environment that would foster recurrence of the tumor. So what we did was to set up a simple experiment to see if some of the properties of cancer cells or non-senescent cells were exacerbated in the presence of media that was derived from senescent cells. Well, these are the models we used. Still, in vitro models use AGS cells, which are derived from a gastric adenocarcinoma cell line. We use MCF10A, which are mammary epithelial cells that were immortalized but non-transformed. And we use palbociclib as our inducer of senescence. And you can see here that after many experiments of optimization of the conditions, we get a nice model of senescence induction using this drug. And 5 minutes? Oh my God. Well, time flies. Okay, so we see that they have different profiles of expression of different of the same bioactive molecules we check in the other set of experiments. We also observed that interleukin-1 beta was one of the main factors that were secreted by these senescent cells. We also observed that this conditioned media derived from these senescent cells were capable of inducing platelet aggregation, especially with media derived from senescent MCF10A cells. So we did a simple experiment. We wanted to check if the media conditioned by senescent cells was able to induce invasion of the non-senescent counterpart. So what we did was also include platelets. At that point, we were interested in testing if there was some synergy between the senescent media and platelets in the induction of invasion. So as you can see here in the black bars, we have the effect on invasion of senescent media, and in both cases, the media conditioned by senescent cells was more invasive, so to say, than the media conditioned by non-senescent cells. Now, in terms of the role of platelets, indeed, there was not much synergy. In fact, platelets, for example, in this cell line was able to induce invasion almost independently of the presence of conditioned media derived from senescent cells. Here, in the other cell line, we observe that platelets actually seems to have an inhibitory role in invasion. So in 2 minutes, I'm going to show you the idea that we are trying to develop right now, and it's more related to immunology. So this is the fact, or this is what the literature tells us. So if you have a tumor, you can treat this tumor with prosenescent chemotherapy, and you will get a tumor that is regressing and is accumulating senescent cells. Of course, these senescent cells secrete things, and this secretory environment can promote the recurrence of the tumor and tumor progression. So the idea is that to be effective, prosenescence therapies must go together with the elimination of senescent cells by the immune system or by other means. So only when senescent cells are eliminated, tumor will regress, and that will translate into a successful therapy. Yeah. So there's this idea then that some senescent cells can recruit and activate components of the immune system, and in that way eliminate or themselves, and in that sense could be something good for the patient, because in that case the tumor will finally regress. So we are working back under this hypothesis that senescent gastric cancer cells driven by CDK4/6 inhibition can signal their own removal by releasing factors that enhance the activities of immune cells. Well, we work with AGS cells because we eventually want to translate this into gastric cancer. What we first did was to identify some immunomodulatory molecules that are produced by AGS cells induced to senesce by CDK4/6 inhibition, and we tested a lot of the expression of a lot of factors that may be involved in cancer progression, and they are a mixture of pro-inflammatory and anti-inflammatory molecules. From this work, preliminary work, it is evident that many of the factors that are upregulated in senescent AGS cells are really pro-inflammatory. But we were also interested in analyzing the expression of some antigens on the surface of senescent cells that will inform us about the possibility that this senescent cell could be targeted by NK cells, natural killer cells, or T cells. So this work has been done by Raul Segovia, who is a PhD student in our lab. So he found that there are several ligands, we call them NKG2D ligands, which are molecules that are expressed in the surface of senescent cells and can be recognized by receptors in natural killer cells. Many of these factors are upregulated in senescent cells. In terms of T cell, we observed that senescent cells, AGS senescent cells, also express or redirect the expression on the surface of MHC1 molecules. So this could also mean that these cells could be targeted by T cells, by CD8 positive T cells. And finally, we did an experiment These are all preliminary, but I think very promising experiments. We wanted to know if the factors released by senescent AGS cells were capable of changing the activation or the populations of peripheral blood mononuclear cells. What we did first was, what Raul did, was to isolate human peripheral blood mononuclear cells, PBMCs, and then incubate those cells for a time in media condition by senescence and non-senescence AGS cells, and then analyzed by flow cytometry the different populations inside within this blood mononuclear cells. So here you have all these experiments were done in Dr. Puente's lab. He separated two populations, one population which is the lymphocyte population, and then separated this population of lymphocytes into those which were CD3 positive or CD56 positive or negative, and he found that there was no difference. CD55 is a marker for NK cells, and CD3 is a marker for lymphocytes. So he did not find any difference when he incubated this population of cells with media derived from senescent cells. So, but so he went on to take another marker, and then he focused only on the population of NK cells, and he found that very interestingly, if you incubate this population here, which is a population enriched in CD55, CD56 positive NK cells, and then incubate these cells, this population with media derived from senescent cells, he found an activation of this population, which was evident by the detection by cytometry of CD107A, which is a marker of activation of NK cells. So when mononuclear then cells were obtained and incubated with conditioned media derived from senescent AGS cells, we observed that the number of active NK cells increased from 43% to 61-something%. This was, of course, determined by cytometry analysis. So this is our work so far. We're trying to test the hypothesis that factors that are released by senescent AGS cells or other types of cells can induce the activation of different populations of immune cells, including NK cells, and of course we have to test if the population of CD8 positive cells is also activated by these factors. And this is, so I would like to thank all members of the lab, Dr. Moore, Dr. Valenzuela, and all students who participated over the years in these projects, Dr. Eduardo Fuentes, Dr. Andres Serrada, who provided us with the facilities to perform cytometry analysis. Some of the funding that we have during the years, and the fund equip, which is, of course, the source of our cytometry facility here in the university, at the University of Talca. So that's it. Thank you. Yes.
Yeah, that's I was two questions. One relates to the concentration of drug you use to produce senescence. So, is it, if you use very high concentration, you kill the cell. So this must be a dose to that.
Yes.
Right?
Well, so that's something we didn't show, but before we set up the next experiments, we always do a lot of optimization experiments. In many of these cases, we also check apoptosis or induction of necrosis by flow cytometry, just to make sure that we minimize the amount of cytotoxicity, but we maintain the amount of cellular senescence. So to reach a certain dose really was a lot of work before.
So the other question I have is, you're only looking at the fact have you considered the possibility of, particularly in the case of Dr. Lewis, cytotoxicity? And do you think that those effects could be mitigated, for instance, by these eliminated by the senescent and the legal cells?
Yes. So, Whitney, who is our student in the lab, who is working actually in the possibility that senescence, endothelial senescent cells could release not only these factors which are soluble, but also some vesicles that could communicate with other components of the microenvironment. Now, regarding the question of cytotoxicity, we know that doxorubicin, one of the main problems that has is related to cardiotoxicity. So, in that sense, it could be that part of this cardiotoxicity is not directly in the myocardium, but in the endocardium, which is basically continuous with the endothelial cells of the vessels. So it could be that then I don't think someone has studied this yet, but it could be that endocardium is really part of the answer of why doxorubicin could have a detrimental effect on the heart. More questions? Yes.
Very interesting. So the last part is very interesting, and I congratulate all of them, because natural killers are very interesting cells. The mechanism by which it kills cancer is very complex. So at a certain point, are you thinking to evaluate how these natural killer cells kill actually?
Yes. Very nice question. Actually, that's the next step. That's the if we reach that step and we can demonstrate that, I mean, Raul can start to write a thesis, because before that probably we are just speculating. So there are some experiments in which you take NK cells isolated, for example from blood, and then incubate them with senescent cells. And by some assay. You can check the number of senescent cells that are killed by natural killer cells. And it has been described, so we haven't done it yet, but it's on the way.